Let's talk a little bit more about the enzyme-substrate complex. So again, the enzyme is our biological catalyst. It's a protein, and the substrate is going to be some kind of molecule that's going to bind to the enzyme. So what's shown down here in this set of 4 reactions, it says here that these reversible reaction steps represent the steps in an enzyme catalyzed reaction. So there's 4 steps that you're going to have to know. The first step involves the formation of the enzyme-substrate complex. So if we look at what's happening in step 1, we have the enzyme and the substrate. So the substrate is going to approach the enzyme, it's going to bind to the enzyme, and then it forms sort of a conglomeration of the two of them, which we call the enzyme-substrate complex. Then what's going to happen is the enzyme is going to put some kind of stress onto that substrate and bring it into a higher energy level that we call the transition state. So it's going to basically start doing a reaction to that substrate in order to get to a transition state. Once that transition state is achieved, in the third step, we're going to get that substrate being converted into product. And then just like we had the enzyme-substrate complex, now we're going to have an enzyme product complex. And you can probably guess what's going to happen in the last step is that ultimately I'm trying to convert the substrate into a product via the enzyme. So in the last step, what's going to happen is that enzyme is going to release the product. So that's going to be the final step. So you need to be aware of all 4 of these steps. So the part of the enzyme that combines with that substrate, it's got a special name. We call that the active site. And the active site has some really interesting characteristics. For example, it has what we call pockets or clefts in the surface of that enzyme. And the R groups from the amino acid residues at that active site, we call those catalytic groups. So those are going to be important R groups for the binding of our enzyme. Also, the shape of the active site is going to be complementary to the shape of the substrate. So the shape of the substrate is going to fit somehow into that pocket or cleft, which will be, again, complementary to the shape of the substrate. The enzyme attracts and holds the enzyme using weak, non-covalent interactions. So there's going to be some kind of intermolecular forces that hold that substrate to the enzyme. And if they're weak, non-covalent interactions, well, what would they be? Van der Waals forces. So we're talking about dipole-dipole interactions, electrostatic interactions, London forces. And again, the conformation of that active site is going to determine the specificity of the enzyme. So what that means is that every enzyme is going to react with a specific substrate. So they will not just be promiscuous and react with any substrate. So this is a little picture just showing you an enzyme, a substrate, and an active site. So an enzyme contains an active site that binds the substrate, forming an enzyme-substrate complex. So here's our active site. Here's our big enzyme. So this big blue, blob, if you will, that's our enzyme. And then the active site, like it said in the previous slide, it's some kind of pocket or cleft here. It's not easy to tell from this cartoon, but basically that pocket or cleft is going to be complementary to the shape of this molecule. So you see it's going to bind in here just right, and then we form our enzyme substrate complex. So after the transition state has been achieved and the reaction has occurred, then we have the enzyme product complex, and what happens is finally the enzyme is going to release the product. Now in this case, what they're demonstrating in this reaction is that the substrate is being broken apart into these 2 molecules, so the green molecule and the red molecule. So again, the enzyme must have put some kind of stress on that substrate in that transition state to break some bonds. So 2 models have been proposed to explain the specificity of a substrate for an enzyme's active site. The first one is what we call the lock and key model, and it was developed by Fischer way back in 1894. So he assumed that the enzyme was like a lock and the substrate was like a key. But what do we know about locks and keys? Is that locks and keys are made to fit exactly with one another. So they're not going to change shape in any way. But the problem with this is that the model fails to take into account the fact that proteins can undergo conformational changes to accommodate a substrate molecule. So what that means is that we know that proteins are not rigid molecules. We know that there is some movement at the molecular level associated with proteins and that they can change their shape slightly. The induced fit model came along in 1958, and it says the enzyme action assumes that the active site is more of a flexible pocket. So a flexible pocket whose conformation or shape can change to accommodate the substrate molecule. So if we look at this enzyme here in blue, you can see that the shape of the pocket, it matches the shape of the substrate to some extent, right? Because we have, there's sort of a bulge here, and then there's another one here. So you can see how those might fit, but they definitely don't fit like a lock and key. But again, we know that this part of our enzyme, or our enzyme in its entirety, can undergo conformational changes. So conformation means nothing more than changing its shape. Now, it's not going to change it entirely, but it can change it slightly or enough to accommodate that substrate molecule. And hence, it's going to fit it just right. And this is what we call the induced fit model, because the substrate is inducing the enzyme to undergo a conformational change.